Okay, so good morning, everybody. Thanks for being here. It is a pleasure to receive all of you to this international conference on systems, on dynamics in systems and in synthetic biology. We will start with a conference opening, and the director of our center, Centro de Recerca Matemàtica, Center for Mathematical Research, will say some words welcoming all of you to this research program that is starting with this conference. Luis, please.
Thank you, Josep. I will be very brief because, as I said, when I am listening to those introductions, I always get nervous. So I try not to make them long. So this is just to welcome you to this activity. As you know, CRM is a research center that it does research, it organizes activities, international research programs in the as a lot of mathematical research centers do, and we try to do well in this sense. For us, it's a very important leg of the center, offer the possibilities to the mathematical community that being together, they develop, they help to push forward the scientific outcomes of the community. So it is our pleasure to do this in fulfillment of our mission. Maybe the thing different in this activity, the current one, to talk about is the fact that it's completely virtual. As all of you know, the pandemic has been terrible in some sense. Now we are endeavoring a new future where we do not know how activities will be. Probably activities, most of them will be a mixture of virtual, of online and semi-online activities or semi-present activities. This one is completely virtual, not the whole program. And to say that CRM has tried to equip and be ready for such things, and we are trying to do well in this format as in the standard classical format. So this is just putting CRM at your service. I enjoy that you, I hope, sorry, that you enjoy the meeting, and it's very fruitful for everybody. Usually I would say, and welcome to Barcelona, please enjoy the city. In this moment I cannot say this, but anyway, welcome to the meeting, and I hope that you enjoy it.
Okay, thanks so much, Luis. So now I will share with you some information. I hope you can see it. Okay, so this conference is within a research program that we are doing at CRM. So CRM typically organizes intensive research programs. These are programs that last for a few months. The program on dynamical systems, mid-systems, and synthetic biology starts today, 14 June, and will end in October 30. And we have different activities that we will do along the program. So the first one is the conference that is starting today and will last till Friday. Then during the whole period, we will do also online seminars, which are free, and you are invited to attend them. So for the online seminars, we will send you the link a few days before the seminar. And finally, we are preparing an advanced course on applied dynamics in systems and synthetic biology in September from 6 to 10. And if things are okay in terms of the pandemic, we are planning to combine a face-to-face option. So people will be able to come here at CRM with all the health measures for COVID. And also we have an online option via Zoom. So you can register either for being here physically or online. So now I will explain each of these events. So I will start with the international conference. So the conference has gathered 32 speakers. We have about 191 attendees. And the speakers come from many different research centers and universities from Catalonia, Spain, United Kingdom, United States, and Russia. Okay, so we have Pompeu Fabra, the Institute of Integrative Systems Biology in Valencia, the Centro Nacional de Biotecnología in Madrid, Oxford University, the three main universities in Catalonia, Universitat Autònoma, Universitat de Barcelona, Universitat Politècnica de Catalunya, the Center for Advanced Studies in Blanes, Princeton University, the CNRS, the Russian Academy of Sciences, MIT, IDIBELL, and the Institute Catalan of Oncology. So one important point that we have been aware in this conference is the gender balance. So we have done our best to have equal contributions from men and women. So for the conference, we have 47% of women speakers and about 53 men, okay? As I was saying, we will also held different seminars. So this will be on Fridays from 12 to 1 p.m. And we will send you the links just a few days before. So the first one will be in 3 weeks, more or less. Then the other one will be in July. Then we have August, that here it's holiday time. And then we will go again with the seminars in September, and in October we will have 3. So this research program, we have tried to do an effort to bring together scientists working from many different disciplines. So the main objective is to put into contact and provide a scientific platform, scientific discussion platform for combining mathematical research, especially dynamical systems theory and mathematical biology, with people that are working in the lab. Fortunately, some of these people have also experience in mathematical modeling, which is very, very interesting. And mainly the seminars will be about a lot of different topics. So for example, the first one will be about complex systems. The second one by Miguel Verdugo will be about ecology of semi-arid ecosystems. Julia will talk about dynamical systems, discrete dynamical systems. Eva will talk about synthetic biology. Marta will talk about citizen science. Sergi Valverde will talk about complex networks and the evolution of technology. And Marc Plana will talk about dynamical systems applied to virology. Okay, so here the gender balance, it's about 43% women and 57% males. And finally, the last event that we are organizing is this advanced summer school. This is intended to be a real workshop. I mean, this will not be a conference or just some people giving lectures. So we are organizing this in order to provide an academic message. So this will be like lectures, but will be very applied. So this will be about mathematics, but we have been trying to provide all the people with tools from mathematics and from dynamical systems that can be used by people doing experiments and interested in dynamics in systems and synthetic biology. So even if you do not have a mathematical background, I really recommend you to attend to this advanced school, because we will try to provide and explain very useful tools, mathematical tools and computational tools for the study of nonlinear systems, okay? So here we have, well, gender balance here was very easy because we have 4 speakers. So we have 2 women and 2 male. So one of the speakers is Mayra Guiar from Becam. She's expertise is in mathematical epidemiology. Then we have Professor Jordy Garcia Halvo from Universitat Pompeu Fabra, who will give us some lectures and teaching biological dynamical systems. Jordy has been working in cell cultures, in quorum sensing, oscillators, and he has a very strong background in physics and dynamical systems, but he's also doing a lot of nice research in experiments, experimentally. Then we will have from Universitat Politècnica de Catalunya, Imma Baldomà and Tomàs Lázaro. Imma Baldomà has expertise in the qualitative theory of dynamical systems, which is a very important topic to understand the dynamics of what we are seeing in the lab or in the field in terms of differential equations. And then Tomàs Lázaro is also has also expertise in dynamical systems. He has been collaborating with us for many years, and he's interested in mathematical biology. And actually, Jordy and Tomàs are giving a talk in the conference. So, well, with these speakers, we aim to cover different subjects from epidemiology, ecology, systems biology, and Imma will provide a more theoretical approach to dynamical systems. But as I said, we have been trying to give you mathematical tools, so things that you will be able to use during your research. Okay, so this is the last slide I wanted to share with you. So this is the schedule of the conference. So the idea is that we will have 2 conferences before this break. So unfortunately, this will not be a coffee break in which we can interact. But anyway, this is what we have now. And then we will have 2 more talks before the lunch stop. And then in the afternoon, we will come back with 3 talks. So the first talk in the morning and the first talk in the afternoon will be for the keynote speakers. This will be a 45 more or less minutes talk. The other talks will be about 25 minutes. And something I would like to say is that if you want to make questions, we will open for questions 15 minutes before the end of the long talk. So that is, for the 45-minute talks, you will be able to ask questions after minute 30. And for the 25 minutes talks, you will be able to send questions after minute 10. So we have thought that the best way is that you can write the questions in the chat, in the general chat, and the chair of the session will choose some of the questions and ask to the speaker. And we are doing this way because probably some of you have same questions, and if everybody is able to see the question in the chat, then we will not have repeated questions. Okay, so okay, that's all for my part. It's almost 10, so welcome again to the conference and to the research program that starts today and will end in October. You are invited to all of the activities. We will be very happy to have you there. And now I give the word, I stop sharing my screen, and I give the word to Professor Santiago Elena from I2CISBio, who will be the chair of the session of today that is about virology and immunology. Thank you so much again, and please, Santi, you have the word.
Thank you, Josep. Juana, are you there?
Yes, I am here.
Fantastic. Good, nice to see you. So, well, as Josep said, welcome everyone to this conference, and specifically to this first session on immunology and virology. We have a very compact program today, very diverse. You take a look at the program. We have from more biological virology side to more theoretical components. And that's a nice balance. I think we all are going to learn a lot today. Now, but one of her main topics is looking for phage in the wild that can be used as therapy against multi-drug resistant bacteria. This is a very, this is an old idea that has been revived very recently or during the last years because the antibody crisis. Right? You all know that we are running out of antibiotics and we need new strategies, and bacteriophages seems to be a very promising one, as I'm sure that Pilar will illustrate during her talk. Okay, Pilar.
Thank you very much, Santi, for the introduction, and thank you, the organizers, and especially to Bifurcations. It's a pleasure for me to share with you some of the work that we are doing in my lab. We are at the I2SOS biome, and well, one of the main problems in global health right now, for sure, is the pandemics of the SARS, but the multi-drug resistant bacteria. As you know, and as Santi said, the use that, or not good use of the antibiotics makes that many bacteria can make some resistant mechanisms that at the end we have a main problem that is a big threat in the global health and is in the world, entire world, in which many bacteria has no treatment nowadays. As you can see here, we have the World Health Organizations that makes global action plans in the Europe or in Spain, in all the countries. There are different action plans in order to combat this antimicrobial resistance. And there are some of the bacteria that are pathogenic bacteria that are considered by the World Health Organization as important in order to combat. Here I just mentioned some of them that are the escape bacteria. Escape group is called because it's composed by Escherichia coli, Staphylococcus, different species, Klebsiella pneumoniae, Acinetobacter baumannii, Pseudomonas aeruginosa, and Enterococcus faecalis and faecium. As many of you know, some of these bacteria are in the normal flora in our body. They can be in the skin, in the gut, in the lungs. Some of them, they also live in normal environment and they are not pathogenic. But in some cases, these bacteria can make infections that can be highly pathogenic, and many of them are now have emerged different resistant mechanisms that makes that there are no treatment and many people is dying due to this MDR bacteria. I'm going to focus in Klebsiella pneumoniae, but firstly I want to do some kind of overview about the problematic in global health. At the right, you have this picture in which normally you put your bacteria in a petri dish, in a lawn, and you have some kind of different antibiotics, and you can see these halos in which the antibiotic kills the bacteria. And in the right, you have what is happening nowadays. Many of the current antibiotics have no action into the bacteria, because bacteria have developed different resistant mechanisms, and we can see in the left. There are different mechanisms in which, for example, they can make an impermeable barrier in which they will not allow the antibiotic to enter into the cell. If this is happening, if the antibiotic enters into the cell, there are different mechanisms in which the bacteria can just, for example, put through the bacteria the antibiotic by efflux pumps. So the bacteria will put again outside the cell the antibiotic, and I will have no response to the bacteria. There are also resistant mutations that could have some different action in blocking the antibiotic action or even drug inactivation. So with all of these mechanisms, and I have to say that some bacteria can exploit different of these mechanisms, we arrive nowadays to the high problematic in which many bacteria have nothing to do with the antibiotics. And this is not only a problem in global health, because if we go in this picture to the right, we have what is happening in humans. We get antibiotics and we were used to take it normally without sometimes a pathogenic bacteria. As you know, sometimes in the past, we were used to take antibiotics for everything. This makes that this misuse of antibiotics, the spread of this emergent resistant bacteria. Some of these bacteria can live in the environment in the hospitals or in medical devices, making this nosocomial bacteria that spreads into the hospital. And many people that get sick and go usually to the intensive care units can stay there and can be infected by these nosocomial bacteria that are high multi-drug resistant. In fact, used to say that right now with the COVID crisis, half of the people that enters into the intensive care units, half of them are dying due to this nosocomial bacteria. So this is a lot of people dying every day without any treatment and asking for compassionate treatments. This is not only a problematic, as I said, in humans, this is also in the environment, and this is why it's important to have a global health approach, because animals are also get treated with antibiotics. They also develop resistant bacteria. This could be, again, go to the humans due to the food, for example, and the same in crops. So this makes that multi-drug resistant bacteria can really go around the whole environment and can make problematic things in all the steps in our ecosystem. So we really need to combat this multi-drug resistant bacteria. I didn't put any picture, but I have to say that in the last years, no new antibiotics have been done. Just modifications of some ancient antibiotics. And also I have to say that sometimes for some antibiotics, emergence of resistance can be acquired in the near months or years after the new antibiotic. So we really need to search for alternative treatments that can stop the emergence of this resistance. And we consider that phages, that are bacterial viruses, can be promising tools in order to combat these multi-drug resistant bacteria. Why phages? Well, phages are the most important component on the Earth. These are just some numbers, but you can imagine it's like 10 to the 7 phages per ml in seawater and 10 to 9 in sediments. So that means that we can just breathe or eat or drink phages all the time. There is an enormous diversity in genomes because they can be circular, linear, RNA, DNA, single, double-stranded, wherever you want or wherever you can think what it can exist in the phages. They have also different morphologies and biological cycles. Just here in the right, you have some pictures. These are the last phages we isolate here in the lab. And as you can see, any of them are totally different. So you can have in the same environment, infected the same cell, different phages with different morphologies. One of the important points of the phages is that they are highly specific. They can be infecting only one strain for a specific bacteria, or they can infect a specific genera, but normally they infect specifically strains. So we can be maybe able to really combat pathogenic bacteria and not commensal bacteria. So this is one of the important points of the phage therapy. And this is something that we are trying to do in our lab. As Antti said, these phages were discovered 1 century ago, and they were already used as phage therapy. In fact, since the discovery, they were used to treat cholera in India, for example, and dysentery, and they had good results. But with when the antibiotics were obtained, let's say that it was better at that point to use something that can kill different bacteria and not something highly specific. And nowadays we are going through the highly specific and, let's say, some personalized medicine. As you say, as you know, sorry, phages can be used for understanding fundamental biological processes. Many of their derivatives as enzymes are used commonly in the labs, but they have also potential application in clinics. And this is something that we are really interested on. Well, phages in biomedicine, and this could be also in, let's say, for animals or for plants, could be used in 3 different blocks. They can be used for sure as treatment, as phage therapy, and it's something that more or less all of you, I think you have heard. Also in gene or drug delivery. In that point, we can put also the phage display that I'm sure also you have heard about. But phages can be used also in prevention and diagnosis. In prevention, we can use them as probiotics, as you say, as you know. As I said previously, the bacteria that are resistant could be spread in the hospitals as nosocomial bacteria, and we can use phages like spraying surfaces or treating medical devices to avoid the spread of bacteria that are resistant. We can also use them as probiotics, as for example, taking some phages or phage cocktails in order to then visit some countries that have a specific bacteria that we don't want to make sick. We can also have phage delivered vaccines. And also we have the potential use of phages as diagnostic tool, because we can use them as a detection and typing of bacteria. As I said, they are highly specific, and we can use as biosensors or bioimaging. I don't really want to enter really a lot in detail, but phages in therapy could be used as phage cocktails. And in here, we can do different approximations, because when you have a cocktail of phages, for example, as I said, bacteria can develop emergence at different points. And we can have different phages that can block these emergent mutants. And we can have already a cocktail that will make easier the treatment. But in that point, we have to do studies of synergies or potential interactions between the phages that we are using in the cocktail. Phages can have also different enzymes that are the phages used to, for example, break the bacteria and go out from the cell. So we can use these enzymes in order to use a different treatment based on phage-derived enzymes. We can also do phage antibiotic synergy. It's also well studied that some phages can be used with antibiotics, and they have good results. As for example, it has been already shown that phages could potentially enter into the cell and can sorry, I have someone outside. In phage antibiotic synergy, we have some potential offers that the antibiotic enters into the cell, and the bacteria try to avoid this entering, removing, for example, some kind of receptors, or the bacteria can avoid imagine that the phage can use the receptor, the efflux pump. So if the antibiotic was removed from the bacteria for these efflux pumps, since the phage cannot now interact with the bacteria due to that, but the phage will have a potential synergy with the antibiotic, because the antibiotic right now will enter into the cell and will not be removed because of the pressure that the phage made. There are no more efflux pumps removing the antibiotic, and now the bacteria is again sensitive to the antibiotic. There are also engineered phages in which you can do chimeric phages or phages as carriers for some drug delivery. and for sure the CRISPR and other methods that can be used to modify bacteria and try to reduce the emergence of resistance. Well, I'm going to focus in Klebsiella pneumoniae. In my lab, we use different clinical strains and different bacteria, but the one that we have done from the last year is the Klebsiella pneumoniae studies. Klebsiella pneumoniae is a gram-negative encapsulated bacterium. As I said in my first slide, is between the escape group. So basically, the carbapenem-resistant bacteria is a priority group for the WHO. It can have it can cause several diseases. And one of the important things in Klebsiella is the presence of an exopolysaccharide capsule. Here in the right, you can see a picture in our lab in which we have in the up the capsule that is, you can see like pink. And then down, you have some of the bacteria in which we remove, due to phages, this capsule. And these new bacteria are avirulent because it is known that the capsule is an important virulence factor. So you can also use phages not only to kill bacteria, but also to remove this virulence factor. And the bacteria will be not resistant anymore. One of the important things in Klebsiella is that there are more than 100 capsular types described. This is making difficult the use of phages because phages, as I said, are highly specific, and sometimes this capsule is the receptor of the phage. So this makes really complex the use or the production of really a cocktail of phages that will really detect and kill all the capsular strains. So our goal is to really be able to create a cocktail of phages that will be able to infect a broad range capsular types of Klebsiella pneumoniae. Well, I put here just a few examples, but right now in my lab we have more than 300 Klebsiella phages. Some things that we are used to do is firstly to isolate from the environment different phages because all of them were isolated here in the nearby to Valencia. Firstly, we describe how the lithic plaques are in the bacterial lungs, and you can see that phages can have different morphologies. They can have some halos, for example, in here, in which you have a highly lithic plaque in the middle, and then it's surrounded by a halo, and this halo is the polymerase activity, as I said in here. This is kind of work in which the phage removes the capsule. The bacteria becomes avirulent, but it's not killed. And then you have other viruses that are really lithic and they have no polymerase, the polymerase activity. For all the phages, we are going to detect and see them by them, in which we can see here that we have different morphologies of the virions. We do as well sequencing of the viruses, and we do here in the top some kind of a spot test in which we will try to have all of viruses. We will test them in different bacterial hosts in order to see the bacterial range, the host range of the phages that we have isolated. I'm going to just mention some of the work done by some of my students. Bea is doing her thesis based on Klebsiella phages. She is using a Genome-Wide collection of more than 100 clinical isolates. All of them, they are coming from hospitals here in the community, in the Valencian community. This collection is representative of the different capsular species, and we have done some kind of sequencing of all the strains, and we have isolated some representative of the clonal complexes of Klebsiella. For them, we isolated 70 phages that are able to infect all of the strains of the 140 strains of Klebsiella that I mentioned, and we really annotate all the genomes. And we are interested in the interactions between the phages and the bacteria. So we understand this dynamic of host-range interactions by spotting. As I said, we do a spot test in the different bacteria. We can do also bactericidal effects in liquid infections and also EOP in plaques. And we have this large matrix with all of the 70 phages with the 140 bacteria, and we can understand all the interactions between them. As you see, you can see that the phages, most of them, they can just infect just one or a few bacterial hosts. But we have some phages, like for example in here, that we have a large infection rate. So these viruses a priori will be interesting for us in order to create our cocktail. We have also Robbie doing another kind of approximation of this phage cocktail. As I said, there are more than 100 capsular types of Klebsiella, but there are 77 reference capsular types. They are considered as reference, and the others are just a few clones that have been isolated in the world. But these reference capsular types, you can book them in the State and Serum Institute, we do it, and we have isolated one phage per capsular type. So right now we have also ongoing work. We have just made the NGS run, and we are doing the analysis of the annotation of the phages, and some of them are really close, as you can see in the two first, in the red and the purple. But there are others that are similar, but with large differences in their genomes. We have found phages that are really small and phages that are considered jumbo phages. These ones that are really long, and we are already doing this analysis. In addition, as we said previously, we are doing the infection range matrix and doing the spot test and the liquid infection to have an overview about what we have with our viruses. In addition, we can also do some kind of biodiversity study of what we can find in our ecosystems. But in here, I think the most interesting point to mention is, since we are with a lot of mathematicians here, and this is something that I want to really check with you, is that we have this kind of kinetics of the bacteria. This is the hours, times, and how the bacteria is growing. And this is done always for the different capsular types. I just put here some examples. Just the bacteria without virus. And then in gray, we will have here bacteria with one phage. In orange, bacteria with another phage, and the combination of two, because we want to see if one phage alone or another phage alone could or not infect the same host. And in the case that both phages can or not infect, if they have synergy or antagonism interactions. And we can see this in how the bacteria is growing. So for example, in the first graph, we can see how the phage in the gray phage is able to kill a little bit the bacteria, but you have soon some kind of rebound of the bacteria, in which we consider that in these cases there are some variants present in the population that already can counteract the phage action and can reach the normal bacterial load. There are other cases, like in the right, in which we can have a phage, in this case in the gray, that can really block the emergence of resistance. This is not really common, sorry. So we will have one phage, that is the orange, that is not able to kill the bacteria. We have the gray that kills the bacteria without emergence of resistance. And we have also the orange or yellow that have similar action like in the first phage. However, to distinguish between these two, if the presence of the second phage, this is always with 3 replicates, I forgot to mention it, it's important to do mathematical models to see if there are differences between the gray and the orange curve. So what we are doing right now is to do studies of the area under the curve. But sometimes this is not so easy to do, if you can see in the low graph, because sometimes you can have growth of the bacteria, then the bacteria starts to be killed and you have a drop, but then the bacteria starts to grow again. So sometimes what we are trying to do in this kind of graphs is to understand how it goes up and down, and trying to do to section the graph in different parts in order to compare the action of different phages. Because in this case, in the capsular type 30, you can see how both phages have an activity, but to see if they are different or not, we really need to go further for the mathematical models and really compare these graphs. In the capsular type 31, you can see how at the beginning, if you do the analysis for the 6 hours, you have a really good lysis of the bacteria, but at one point it starts to emerge bacterial emergent resistance that in some hours they can reach the normal growth of the bacteria. So in here, I just wanted to really show you that we have different forms of growth and different dynamics of interaction between the phage and bacteria, and we can really go further to understand differences between phages, to understand interactions between them. And one of the important points is how to understand the blocking phage-resistant pathways in Klebsiella. This work is done by Lucas in his thesis, and we are really trying to obtain resistant bacteria. We add firstly one phage that we know that will kill the bacteria at the beginning, but then bacteria rebounds fastly. In here, we took resistant bacteria, and in total, we have isolated right now 120 clones. And what we do is to again infect the culture and to see how somehow the bacteria will again make resistance. So as you see here, some viruses, how they have different profiles, and for some phages, they at even 12 hours they have blocked the growth of the bacteria, but somehow at one point some resistant bacteria emerge and again go up. So still resistance occurred, and what we are doing is going for the F3 and took again some phages that will kill this bacteria, and in order to create a cocktail that will block for the first all the emergence of resistance. So we have two approximations. We can have original phages and original anti-resistant phages that we can add firstly in our cocktail and try to reduce the emergence of bacteria. But what we have observed until now is that even if we do that, at one point bacteria emerge resistance. And we can also do some kind of anti-resistant phage that will be introduced later in the culture. And this could be in the left because the resistant bacteria are already in the population, or in this case in the right because they are bacteria that are emerging during duplication of the bacteria. So our idea is really to combat this emergence of resistance with different cocktails that will be able to kill the bacteria and to have different receptors in order to reduce this emergence of resistance. But as you can see, there is still a lot of work to do. And just to mention some works that have been done in other phages and other models with bacterial phage dynamics, some kind of studies with co-infection or some mathematical models for evolution. And there are others that can be also in a spatial way with the reaction diffusion models and how the bacteriophage could spread in, for example, in a biofilm. And for example, in here, I will mention how they can measure the phage decay or the phage stability in time in the absence of host. So it's just phage stability. We have been done also this for some of our phages. And we have to mention that it also depends a lot of the conditions, for sure, temperature, humidity. So it's difficult really to measure in real conditions. There are other models that can also make approximation how the phages can bind to the bacterial host and how they are afterwards are released after lysis of the cells. And there are different curves that can be fit for the growth of the phages in different bacteriophage host dynamics. So what we do is experimental evolution. So in one point, as I mentioned, we are just picking phages, hunting phages from the environment, trying to have phages that are already in the environment in order to kill bacteria and the emergence of the resistant clones. But we are also doing ongoing experiments of experimental evolution. Here I just mentioned one work that we did some years ago, in which we try to get evolution of a phage in experimental conditions in order to have a phage that could make better lysation of the host. And if here you can really clearly see how the founder phage have small plaques, and how in 20 passages the evolved phage evolved with higher lysis plaques. So it increases lytic activity. But we are also interested in increased infection rates and, as for sure, reduce the resistant emergence. And I want to thank my collaborators, especially Rafael San Juan, and all my students and technicians that work hard to do this work. And I want to for sure to acknowledge the Smith who funded all the work, and all the for my Cajal, and for sure the hospitals that helps with the strains. And thank you very much. Do you have any question?
Thank you, Pilar. That's a really nice talk. Really, really nice. We have a couple of questions in the chat. One is from Josep. He's asking whether if you do the experiments in an in vivo system, in an animal, with its own complex microbiota, is it possible that the phage can somehow evolve and lose a speciality and then infect other non-pathogenic bacteria?
Well, this is something that we try to do in the lab firstly. So we are in contact with hospitals asking for highly pathogenic bacteria. And for example, in Klebsiella, there are high-risk clones with this carbapenems activity that we are trying to get to, let's say, evolve the phages to really infect this bacteria, but not the Klebsiella that are commensal. Because if we compare with antibiotics, for sure, we have phages that are highly specific for Klebsiella. So at least this is a good point already. But we are trying to be able to really detect and kill only pathogenic bacteria. This is something that we have to do firstly in the lab. And then there are animal models. Firstly, normally in phage therapy, we use Galleria melonella, it's a Mosaic larvae, and then we can go for mice or rat. But this is something that we have to do firstly in the lab. But as I said, phages are really highly specific. In Klebsiella, as I said, we have more than 300 phages. And I can tell you that mostly 90-95%, they infect only one capsular type. So we are trying also to do recombinant experiments and evolutionary experiments with different species to really have increase the range of activity, because we right now publish there is only one phage for Klebsiella that is able to kill 9 capsular types, and we have already some phages that kills more than 20, and these are natural phages. So we consider that if we can make some experimental evolution of these phages, we will be able to infect a higher range. And it is known that some highly pathogenic bacteria are specific capsular types. There are 5 capsular types that are the more than 50% of the high-risk clones. So maybe with a cocktail against these high-risk clones, it will be interesting and will not kill other bacteria. And it's not so easy to evolve the ability to kill another capsular type. It's not so easy. Just for a few host range, wide host range phages.
Okay. Juana is asking whether in Spain the use of bacteriophages in clinical settings has been authorized.
Well, it's something that I said also, no, I didn't say it, but I am member of the FAGOMA, this is the National Network of Phages. And we sent last year a document for the AIMS in order to regularize all the use of phages. Right now, in Europe, it's not possible to use phages as a common treatment, even not in the US. But since the last 5 years or something, we can use phages as compassionate use. I mentioned it that right now for the EQ patients in COVID, half of them are, the death of the half of them is due to nosocomial infections. Right now, and since December last year, the FDA in the US authorized the use of phages for COVID patients in the UK. So we are getting again to the AIMS with this information, like, please, we have to do that also in Spain. And right now, we use also them as compassionate use. I know that in Vall in Barcelona, it is used for mycobacterial infections. Here in Valencia, there is in La Fe one team that I'm working with them for another mycobacterial abscessus infection that we are searching phages to use them as compassionate use. 2 weeks ago, I sent to Madrid again another phages for another E. coli infection. That this is important because it's an E. coli bacteria that we have them everywhere in our body. There is a patient that has a prosthesis in the leg with infection with a specific strain of E. coli, and the girl is going to lose the leg. So in Madrid, they tried to find phages. They didn't get nothing. The medical doctor sent me here in our lab the bacteria, and we found five phages that I sent 2 weeks ago, and they are going to try, and it's going to be used. So right now in Spain, if you have some case that you know someone that has the problem of a multi-drug resistant bacteria, there are some labs that we are trying to search for phages to use them in clinics.
Okay. Just another question from Anel Nurtai. She's asking whether in the curve that you show that there is a rebound of the bacteria after some time. She's asking whether there is or you are doing studies, or you know something about how the initial dose or the initial concentration of phage will control that process. Maybe there is a possibility that, or there is a situation where a given amount of phage is enough to stop reduce the likelihood of the bacteria to become resistant.
No, it's good. I mean, nice question, but no, we try to do it with different amounts. We start with different MOIs, and no, there is always a range of MOIs that are good for infection. If you add too low, you don't have enough bacteria to bacteria growth fast. If you add a lot of viruses in the beginning, bacteria die and you cannot even recover viruses, but you have always rebound of emergence of resistance for phages. And we try with different, yeah.
Okay, and there is a question from Alfonso Jaramillo. He says that what is responsible for the intermediate optical densities observed in the plots of K13 and K30?
What?
Right, you need to go back to the slide.
Okay, I'm going. Okay, in here?
Yeah.
Which one? And you mean this thing, you mean?
The K13 and K30, but no, yeah, probably, yeah, that's that rebound, yeah, that peak, I guess. He refers to that peak, I guess.
Yeah, I think it's the bacteria start to grow. The phage is not, it needs time to start to kill. In that cases, I think is the phage that needs more time. And once the bacteria start to grow, the phage, when it acquires a good concentration, that you can see that the lytic activity starts to go down the effect of the bacteria. But this is normally, we are used to see this.
Mm-hmm. I hope that was the right answer, question. But anyhow, so if let's see, we don't have.
In the long term.
In the long term, yeah.
Well, why it rebound in the long term? Look, I'm going to show you. You mean why it not rebounds in these two, or why it rebounds here?
It doesn't increase.
Because in here, in the 31, it rebounds, but it's because at one point some clone starts to grow, and it takes long firstly to appear, and secondly to grow. And in here, in the K13 and K30, we don't see really an increase. It's already blocked at 16 hours. Why doesn't it increase? Okay. Because we have the two different phases that in that case, or in this one, that the bacteria is not able to emerge a resistance. At least in this time, we don't know if we leave it longer, what will happen. But normally what we have is the K32 type that infects nothing, or the K31 in which you have a rebound. But in sometimes you have like K13 or K30 in which bacteria are not growing, because the phase in the case of K13 is only one phase, the K30P 2.2, that is able to really combat the bacteria. And if you see in the orange, the presence of the other phase, the K7, do nothing. So only one phase is capable to really avoid the emergence of resistance. And in the second case, in the K13, you have both phages that are able to really block the emergence of the resistance. So in that period, the bacteria is not able. And these phages are phages that are host range. The host range is high, is wide. So the phage can have different mechanisms to really maybe or the receptor, or maybe how they kill the bacteria. So for the bacteria, it's difficult to emerge a resistance.
Yeah, okay, Pilar, thanks a lot. Thanks everyone for the questions. We are going to make a break now. We are back at 11:45, so in 17 minutes. Next speaker will be Saul Ares. Okay, so I'll see you all in 15 minutes. You all, okay, thank you, recording in progress, boys. Assume you are all back. We move to the next speaker, which is Saul Ares from the Centro Nacional de Biotecnología in Madrid. We are moving from a more experimental biological side to a more theoretical one. And Saul is going to tell us about predictability in the coronavirus, I assume, during the coronavirus pandemics. Saul, whenever you want, you can share the screen with us.
Thanks a lot. Share screen. The desktop, I guess.
Yeah, it is.
Okay, full screen. Okay, there it is, right?
Yeah, perfect.
Okay, thanks a lot, Santi, for the introduction and also for the invitation to talk here about my work. And yes, our title is Predictability. Can the turning point and end of an expanding epidemic be precisely forecast? Which is a little bit of a wink to a classic paper by Lawrence of the Lawrence attractor fame that started a little bit the modern field of chaos, and had a similar title, but about weather forecast. And we will see that we will come to similar conclusions to which he found back in the 60s. This is joint work with Mario Castro, Jose Cuesta, and Susana Manrubia. So, well, it's me speaking today, but it's the work of all of us. Okay, so generally about epidemics, we know that the analogy with weather is interesting in lots of sense. One of them is that with epidemics, we know that they are like extreme weather events. We know that eventually they are going to happen. We know that eventually a new epidemic will come, but what we don't know is exactly how and when and how big it's going to be and how important. And that's about the setting of epidemics in general. But then we have the question of, even when we are inside an epidemic, how much can we predict about it? We remember March 2020, when it was clear that this was a big world problem, and models started publishing predictions everywhere that made it in the news. And you know that some months later, the news is how miserably most of those predictions failed. So why did they fail? What are the problems with predicting the dynamics of contingency in an epidemic? First problem is the quality of data, that is retrieving good enough information. You might remember that it was almost universally that protocols of how data was obtained were changing from week to week, making numbers difficult to compare from one day to other. Also, much later, we learned that actually the data was not correct, that lots of things were missing, that it was very difficult in real time to really know what was happening. So no matter if you have a very good model, if you feed garbage to it, the output will be garbage. That's the first problem. But another problem is, of course, the quality of models. Even if you have very good data, you need to have very good models if you want to do something with it. And that's a little bit more what we're going to speak today, but only very, very slightly, because the real problem is about the intrinsic dynamics of this kind of processes. And what I'm going to try to convince you is that by definition, they are very difficult to predict, no matter how good your model, no matter how good your data. So is forecasting doomed after a few days? Let's see what we can say. So the thing is that the mathematical framework in which these predictions are done has evolved a bit. It's a bit more sophisticated, but is mostly still solidly grounded on work coming from 1927, when Kermack and McKendrick proposed their famous SIR model, which is very simple. In essence, you make compartments with the population, and you put all the people in a given population that you're studying, that can be a region, a country, whatever, in one of these compartments. The simplest case that they formulated in year 27 had 3 compartments. One for susceptible people, which are those that haven't been infected yet, are healthy, and are, as the name says, susceptible to infection. This next compartment are the infections, that is those infectious, the people that are infected and at that moment can produce contagions on other people. And then we have a third compartment that we call R for recovered, but that actually includes people that are recovered and that cannot infect anymore, but it's also where we include the deceased people that unfortunately also cannot infect anybody anymore. And the dynamics are extremely simple. The dynamics come from basically what you would have in simple chemical reaction models, and applying the mass action law, it's exactly the same. So the number of newly infected people is proportional to the number of already infected times the number of susceptible, and then you have a rate at which the infected get recovered. And that's it, that's all. This very simple idea is what is behind all the models that are being used today. Of course, what you can do then is add more and more complexity to this. You can add more compartments. You can add compartments of people in hospital, compartments in people in ICU, compartments of people that have been infected but are not infectious yet. You name it. What happens here? The old school way of modeling this is using ordinary differential equations, which by definition are deterministic models. So you have a model, you have some parameters, and you have an initial condition, and that will give you the whole trajectory of the dynamics for forevermore. Fortunately, this is changing, and more people are using stochastic models where the role of fluctuations is taken into account, and that's especially important when we're talking about not so huge populations. That is probably where this kind of models make more sense. So should we say that these models are all bad? No, they're actually very useful. Even the simple SIR model has shaped the way in which we think about this problem. For instance, the concept of herd immunity comes from the SIR model, because the growth of recovered people doesn't get to 100% in the model, depending on how infectious the disease is and on the initial condition and on several things. There will be a point at which the infections will go down. And also the concept of R0, the basic reproductive number, also comes from an analysis of this very simple model. So there's a combination of parameters, which here is very simple because it's just how infectious the disease is over how quickly the infected recover, which tells you if the epidemic can grow or not, depending on if this parameter is bigger or lower than 0. So at least for setting our framework and letting us think on how the epidemic evolves, this model has already played an important role. Now, the problem with it, let's go back to this slide. The problem with this kind of model is that it's so simple that it only has one possible mechanism for the infection to stop, which is reaching herd immunity. The only thing that changes with the different parameters is how quickly or how slowly this will happen and how high the threshold for herd immunity is. But eventually you have to reach this threshold before the infection stops. And there is no other way. There is no other mechanism by which this simple kind of model can let you do that. Of course, if you change parameters into it, for instance, you say you start a lockdown, so your infectiousness reduces, and you reduce that parameter, then things change. But still, it's always a herd immunity mechanism. What is happening there? So we thought, okay, there's lots of people doing very sophisticated models trying to do predictions, but we want to try to understand what's going on. So we want to try to understand how a lockdown, how non-pharmaceutical measures, actually, what's the mechanism by which they can help stop or not one of these epidemics. So to do that, we went basically to the next step. We built a model where we took the simplest possible, the SIR model, and we added just one compartment. Okay, I have 5 compartments drawn here, but you see that D and R are the deceased and the recovered, that we just differentiated them here in case we could use the different data for deaths and recovered to try to fit data, but it's the same. The real new compartment here, do you see my pointer? Anybody out there? Do you see my pointer?
Yes, yes, we see it.
Great, then I use it. The real new thing here is this compartment C, which we, at the beginning, we termed it like confined people, people are under lockdown. These days, I prefer to call it careful people. So we basically divide the susceptible population now in 2. The old school susceptible and the new C compartment of careful people. And they have different rates of getting infected. The simplest thing is that the careful people cannot get infected. But more generally, you could also put another arrow here from C to I with a different beta that means that these people would get infected at a lower rate. This is work in progress, but I can tell you, spoiler, this is not going to be the story today, that doesn't change anything. That just changes the value of the parameters you will obtain when fitting data, but doesn't change at all the behavior of the model. So for simplicity, we just assume that C do not get infected and only people in the S compartment can get infected. And the fun thing is that being in S or C is a dynamical process. So with a certain rate Q, people that are susceptible become careful, they stay at home, they use masks, they do all they can to try to not get infected, and with a certain parameter, a certain rate P, they become susceptible again. which means that they get tired of all of it and are not so careful anymore, or maybe they are essential workers and have to get exposed no matter what, but they are again in the more exposed group. And just with that, this very simple change in the model, you get new mechanisms and new phenomena going on. So the equations are not just now like that. The only change here is that we have this very simple linear terms to change in between S and C. And the thing is that we can do some maths. The thing is that not only we can do simulations and stuff, but we can do some things with paper and pencil, which will help us a lot. That is, assuming that the infected people at any moment are much lower than the total population, which is something that definitely happened in the first wave of COVID, for instance. This is a seroprevalence study for Spain published in the Lancet that showed that by end of April, May 2020, the seroprevalence was 5%. That is, only 5% of people in Spain were infected by that time, which is a lot, but still is very small compared with the total population of the country. So this kind of assumption is rooted in reality. So when you do that, then doing maths become very simple, and you can actually obtain a closed formula for the number of susceptible at a given time and how that evolves in time, and the same for the number of infected. And funny enough, it depends exponentially on something that also depends on time. So it's kind of an exponential. And if you look here, the exponential of an exponential, because we have another exponential here. But the fun thing is that this R0 star that we called it is how R0 basically evolves in time. At time 0, it is this beta over R plus mu, whatever, which is the R0 that we know from the SIR model, but then it evolves in time. And the interesting thing is that the long time behavior of the model will be given by the asymptotic behavior of this R0 magnitude. If this R0 at infinite time is bigger than 1, it means that your lockdown is not effective. That what you will find is that at the beginning you have an exponential. Note that here this is semi-logarithmic axis. At the beginning will have an exponential growth of cases, just as given by the SIR models, but then they will slow down, but eventually get to another slower, still exponential growing phase until eventually you reach herd immunity, and then there it will stop growing. And this can be misleading if you are looking at the data and trying to know what's going to happen from counting cases in the real world, because you can think that this is slowing down means that your measures are being effective and you're going to reach a peak and start reducing cases, and it might happen that you're just slowing down into a second growing phase where you are still growing, and that's not enough. And that's given by a very simple ratio of the elemental parameters of the model. And if you look at the real data that we had back then, that remember is not very good data, but is what we had, you do see that you could fit, let's say, an exponential there and another exponential there, and say, okay, why isn't this crossover from one behavior to another? And it's clear that we are still very far from herd immunity. So why is this going to start growing or not? And then from March 28, you have another behavior. So what's going on here? And the thing is that when this R0 at infinity star parameter is smaller than 1, that means that then you will have a crossover between two regimes, but then the second regime will be a decreasing regime. That is what you want. You want your measures to be so effective that you stop the increase of active cases and you eventually go down. So, and for that, you basically need Q, the parameter of how many people get careful, or how fast they adopt, how faster they are going into the careful case as compared to leaving it with a parameter P. You want this Q to be big enough, then this will be smaller than 1. And then that means that you will effectively curve the dynamics and reach a peak and reduce what's going on. So this is great. And actually, it's what we finally observed in the first wave in Spain and in lots of waves all over the world, how strict measures like strong lockdowns curved the dynamics and allowed for improvement. But now, since we had this model, we thought, okay, let's go and try to fit it to data. And we tried to, when we tried to fit it to data, we found that this was very difficult. We found that different sets of parameters will give us equally good fits. So we thought, okay, there's a problem here. We can't really say anything. So long story short, we went into a Bayesian scheme to fit the data, where you basically get the distributions for each parameter. And here you see the distributions that we obtain for each one of the parameters. And this tells you a lot about the uncertainty of what's going on. You already see how these different parameters can have different values with different probabilities that are consistent with the trajectory of the epidemic that you're observing. When we plot that against the data, we obtain these green shadows that I'm showing here. Both curves here are the same. This is just the linear scale, and this is in semi-logarithmic scale. This is the number of active cases. This orange curve would be the median of the curve obtained using the median of each of the parameters and integrating the model with that. But this gray shadow is where you have the 95% posterior density. That is 95% of the fits that you obtain are here. And you see that they can do very different things. Some of them keep growing forever, while others will reach a peak and go down. So at this point of the data, you don't even know if the epidemic is going to grow exponentially forever, or if it's going to be controlled. So you don't even know if you're going to have a peak. It's not that you cannot make a clear quantitative prediction of what will happen, is that you cannot even know if something as important as reaching a peak or not is going to happen. That would be a controlled epidemic, and which is the case with R not infinite smaller than 1, and this will be exponential growth, which is what will happen if that parameter is bigger than 1. And not even that we can determine with certainty. And just 1 week before the real peak happened, analyzing this data with this model, still anything could happen. Actually, this thing here is the probability of reaching a peak. And from the first case, and you see that there is a different probability of reaching it, like 50 days after the first case, 60 is the peak, 70, 80. But the thing is that integrating this histogram here, this is more or less 25%. That is the probability of reaching a peak 1 week before we actually saw it was around 25%, which is not that much. Fortunately, then it happened. Now, how does that look? So we start with a very few days, and now I'm going to be fitting the model, adding new points, and each day fitting the model. Here we have the predictability interval in gray. The orange is the curve that you obtain with the median of the parameters. And here I'm going to show how the distribution of the different parameters evolves with time. And you see that at the beginning, some of them are even flat. You have no idea of what's the value of that parameter. After some days, the model is very clear that this is going to grow, 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 and grow. But as time advances, things started to change slowly, but suddenly, you see, from this day to this day, we have a qualitative change. Here, the model is already not so sure that this is going to be growing forever. And we see these peaks here appearing in the values of the parameters. That is, suddenly there are some values that tell you that things can be different and that this can be contained. And as you go on, you see that we get to the prediction that we saw before, where everything can happen. And if we continued longer and longer in time, eventually this would collapse into a peak that goes down. Should we blame for this the lousy data? You see here how jumps in the data show you that things are very crazy. These are changes in protocol of how you count deaths or infected cases, or everything. Is not that the real data was doing this, but this is the one that you have reported. Actually not, because what we could do was to use mock data. So we took the equations, we gave them a fixed value of the parameters, we produced perfect data by integrating them, which is this perfect data. And when you try to fit that with the Bayesian approach, to find that even with perfect data produced by the model and fitted with that very same model, you still found the same problem. That after some days, you don't know if you're going to have a peak and go down, or if this is going to grow forever. So how can we fix this? Using better models. We will see, we are working on the generic point of models and see what they can do, but I think that this adds even more problems to predictability. I'm not going to tell more about that now. Using more data, this can definitely help in the short term. For instance, here we have Google retailing recreation mobility that we know that in red, and it correlates very well with the weekly variation of new cases, especially if you delay it by 14 days. In gray, you can see here that the correlation is very good. So you can use that extra information for making predictions, but still you have a very limited time framework. So with that, I basically want to finish. We have dynamic unpredictability in this kind of system, which comes from basically the fact that it's an exponential growth. When you have dynamics that are exponential, the problem is that your errors also grow exponentially. It's not chaos like in weather forecast. Here you have a more simplest case, but still exponentially growing errors for exponentially dynamics means that after some days, a short period, you really cannot say what's going to happen. So we will have to get used to work more like people do in weather forecast, using probabilities and the probabilistic predictions, and also learn more about them on how to formulate our models, on how to obtain data of better quality. And still we have a problem, that is the dynamics of the epidemics depend on the dynamics of decisions that people make, which are very, very difficult to study and predict, and in weather they don't have that problem. So with that, I want to thank you for listening. In this link down here, you can download, if you want, scripts that we use to analyze model with the data with this model and stuff. And well, thank you all.
Thanks, Saul, for such an interesting presentation. Actually, we have a number of questions waiting for you. The first one came from Giuseppe, and he's asking whether time-dependent parameters that are not captured by the autonomous differential equations may make uncertainty even higher.
Well, I mean, definitely, if fitting parameters that are fixed in time is difficult, fitting parameters that dynamically change is even more difficult. That's one strategy that you can use, of course, using a kind of simpler SIR model and changing parameters with time, or using more sophisticated models with time-varying parameters. But that definitely it's not going to help you with this intrinsic predictability problem. It can help you to predict the past very well, to fit your data very well. But what you want to do is not to predict the past. Predicting the past is easy. What you want to do is predicting the future. Predicting the future is very difficult. And you don't even know how those parameters are going to change in the future. So it doesn't really help you overcome this problem.
Right. So another question from Rubén Pérez Carrasco. He's asking, well, actually he said, can we analyze the profiles of uncertainty at long times to predict which measurement, if done properly, will maximize the prediction of the evolution in time?
Well, yes, I guess you could. That's a very good question and a very good proposal. But but from what we are studying now, the work in progress that we are doing, it seems very difficult. So trying to get more careful or more detailed in your modeling or in looking at the different fits that you obtain and trying to see what's better doesn't really seem to help much in knowing which will be the best choice.
A question from Conrado Catarcione. He is asking whether the model can predict a second and third waves.
Just as it is, it really doesn't. As it is now, it's a one wave model. Let's say it grows forever, or it reaches a peak and it goes down. Of course, what we know is that there are lots of things that are related to this appearance of second waves. One is, for instance, that the behavior of people is different in different times of the year. This is, for instance, COVID is a respiratory virus. So if people spend more time in insides, then there are more chances of contagions. Another are the dynamics of people themselves. I strongly recommend you regarding this to check out new work that Susana Manrubia has just posted with Damian Zanette in the archive, where they do a stochastic model that includes dynamics on how people react to the fact that there are more infections around them and makes them more careful or not. And when you take that into account, when you take into account the dynamics of the reactions of people, then you do have multi-wave as a result. In this very simple rule of 4 that I told you today, that still doesn't appear.
There are more questions, but I'm going to summarize 2 questions together. Coming from Manuel Matias and from Mark Plana, that basically is about, this is a SEER model, which is of course a well-mixed model, is a mass action. And the question is, do you really think that that's realistic if you try to apply to a nationwide level, and whether there is a trade-off between making the more structured models and the predictability?
I mean, sure, there are 2 aspects thing. When you try to use one of this well-mixed kind of mean field model, you have to be very careful which you define as your epidemiological unit. If you think of a country like, for instance, the United States, it's clear that the situations in different states were very different. Even in Spain, we've seen after the first wave that at different time, different regions had very different dynamics. So to which point you can put it all together into a simple model is something that you definitely have to discuss. You have to be very careful what you define as your population. And then there's the second part that, of course, you can be more careful and take into account the structure of population, structure of contacts between people a little bit. You can do that in a mean field way, or you can do that using a network analysis. We're actually doing that now. We're working on understanding, again, not predicting, but understanding what are the effects of vaccinating population. And for that, we're taking into account this kind of analysis. And the thing is that we don't think that this changes in any way the conclusions that we reached here. That what you gain in realism, you lose in a more complicated model with more parameters, making things more difficult to find out.
Yeah, there's a last question here. Okay, the last one from Jay Barrow. He says that you are always considering linear deterministic model for the Bayesian estimation of parameters. Do you think this approach is enough to capture the uncertainty due to the heterogeneity of the data? For example, the relevance of unpredictable superspreading events?
This definitely can be done much better. I totally agree that this dynamics of superspreading events is not built in this model as it is, but basically just plays in favor of our conclusion, because superspreading events make it even more unpredictable what's going to happen. So if in the simplest deterministic possible case, things are unpredictable when we take into account these details of the dynamics, which are even more, let's say, bursty or stochastic in some sense, things are not going to get any better.
Yeah, okay. So thanks, Saul, again for the talk and all the discussion. Thanks, everyone, for the questions. So we move to the next speaker, which is Ana Maria Bukovic from the IHUSIS Bio. And she's going to present her work on genome association studies. She will just present this technique to people in the audience that might not be familiar with it, and how it can be used to identify genes that are relevant for viral infections.
Okay.
So whenever you want, Ana Maria.
Hello, everybody. Thank you for the introduction, Santi. I will be talking about genome-wide association studies and how you can use them to maybe understand better how certain hosts respond to viruses and virus infection. So to start, I will first say something about the background for genome-wide association studies. So in this picture, you can see like a cell and its genome, which are the chromosomes. And then here in one chromatid, you can see what this actually is consisted of. It's genes and intergenic regions. So genes are the coding part of our genome, and intergenic regions are involved in different control of expression, for example, of the genes. When we talk about single nucleotide polymorphisms, we talk about these one nucleotide changes in the genome that can happen in different positions. You can have homozygous changes, which is the same letter, or heterozygous changes, which has a different letter. SNPs are important, or these single nucleotide changes, because some of them can cause pretty serious diseases like rheumatoid arthritis, breast cancer, Alzheimer's disease, or Crohn's disease. So they are very interesting to study. A method that can understand and connect better the disease with the genotype is a genome-wide association study. In this study, you have a huge variation of genetic variants in a population. It can be a human population, plant, an animal, whatever you can genotype. And then you perform certain statistical analysis, which give you p-value, or how well this one change in a nucleotide is associated with the phenotype you're observing. So this method is really good because it can connect the phenotype and the genotype, and you can have an easier way to see what is the genetic background of a certain disease that you are interested in. All these nucleotides, you can obtain information about them or the genotypes by sequencing, which is really available these days. So why would we want to do a GWAS study? Well, first of all, it has proved to be very valuable within the last 20 years. Since sequencing prices are dropping and sequencing is more available, you can genotype a lot of individuals very easily and obtain a lot of information. And then you can perform your genome-wide association study by phenotyping for whatever trait you are interested, disease, height, weight, heart problems, whatever. And you can identify certain genes that might be causing this. And further describe like a network, for example, different genes that might interact with this one, what happens in the cell, for example, if you perform certain studies, if you remove this gene, or if you change the expression levels. So it's a very good method to connect diseases with genotypes. Different successful studies that are done with GWAS. For example, a lot of cancer studies have been done. In plants, you had root length studies that were very successful. Diabetes, for example, pharmaceutical companies use GWAS very basically daily to test how certain populations might respond to certain medications. So in our GWAS study, we wanted to study targets of viral adaptation. So we work in a plant. Our host was Arabidopsis thaliana. This is a very well-studied model organism. There are a lot of different genotypes, as you can see in this picture, just by size alone and leaf shape. you have very different variants. There are over 2,000 different accessions described or different genotypes. It's well sequenced, well described. The genetic regions are well described and annotated. So it makes our life a lot easier if you want to do a GWAS study. Also, this plant is really small and easy to handle. So this also makes it quite accessible for studies. The virus we used was a potivirus, Turnip mosaic virus. So this family of viruses is RNA, single-stranded RNA. And they infect various types of plants, mostly brassicacea like cabbage, kale. They also infect Herbacea thaliana naturally. And they do cause a lot of damage in agronomical studies. So we use two different strains. We use an evolved and naive strain. So a naive strain, or the ancestral strain, here called TUMV-AS, was basically obtained from Nicotiana bentamiana and mechanically inoculated by causing damage on the plant. And after it produced symptoms, we saw in a tissue that was infected, we collected it, and this was the naive virus in the study. For the evolved virus, we took the ancestral virus and infected Herbacea thaliana. Afterwards, we did passages, or basically we collected infected tissue, prepared it for another mechanical inoculation, and then infected new healthy Arabidopsis thaliana, waited for symptoms, collected infected tissue, and did this repetitively for 13 passages or 13 steps. And this was our evolved virus called TUMV-DV. So what we wanted to measure in our GWAS study, we wanted to measure infectivity, or percentage of infected plants per inoculated plants. We wanted to monitor disease progression curves. So this is the development of symptoms through time, where you take the number of plants that are infected each day through 21 days, and then you use a formula that calculates this and sums it up in one value. Then we wanted to phenotype symptom severity. As you can see on this scale, we wanted to phenotype how severe are the symptoms in each genotype of our plant. So we have 0 healthy plant, 1 plant that is much smaller and doesn't look as healthy as the uninfected plant, 2 where we see some yellowing in the plant, but not very severe, 3 the yellowing is much larger, 4 the yellowing is basically covering the whole plant, and 5 when the plant becomes necrotic, which basically means that the plant has died. We also phenotype binary data like necrosis and resistance. So basically necrosis if there are dead plants or not. 0 is if there are no dead plants, 1 if there are. And resistance means that a plant hasn't shown any symptoms, or it was not infected. It did not show symptomatology. This was also binary. 0, the plant was healthy, and 1, there were sick plants. So what we did an association analysis with the help of linear mixed models. These models are widely used in genome-wide association studies, and they try to find the specific SNPs in the genomes associated with the traits that we are looking at. So we used a software called Limix. There are many other softwares you could use, but this was available for our study, and it came from the laboratory of our collaborators, so we decided to use this. So basically with the linear mixed models, you fit a mixed or a univariate linear mixed model, and you look at a single phenotype, like only one phenotype, for example, necrosis, or you can look at multiple phenotypes at the same time. For example, you can take necrosis, disease resistance, symptomatology, and do a study at the same time. You don't have to do it separately. So these models are very good because they can account for certain random effects and certain noise that makes the study difficult. So to take a look at the model, basically this is our Limix model over here, and it's separated in fixed effects, this, or random effects, which is this. So in fixed effects, you will account for example, SNPs in the genotype and covariates. Covariates can be age, ethnicity, sex, so certain things that might influence your study. As you can see, we have the sample design. The sample design here is the genotypes. We account for that in our model. The trait design is basically the phenotypes you are looking at, and the effect size. The effect size is how much does a certain SMP in the genome cause the phenotype? How much does it, for example, if you have from 0 to 100%, does it cause 50, 90, 100%? And in the random effects, you account for things like population structure, which I will explain in the next slide, certain noise, all the things that might cause problems in your model. So in here you have variation in trait to trait. So basically between your phenotypes, and in here you have sample to sample variation, which is basically your genotypes. So you account for that in your model. There are certain problems in the Geos study that are well known, and they are measures have been taken to account for them, for example, by using linear mixed models. The biggest problem is population stratification. This causes false positives. Population stratification is basically if you have two subpopulations that might live in a different geographical region, due to mating that is not random, you might have a higher presence of certain SMPs in one population and in the other, but they are not directly correlated with your disease. But if you perform the statistical analyses, these differences will pop up probably, and you will think that this is associated with your trait, but it's just a difference between the two populations. So you account for this in your models. A nice way to see if you have this problem before you do your study is to look at the quantile plots. So in here, for example, you will account for the theoretical distribution here, and the thick line that we see is the actual distribution of your traits. As we can see here, all these pretty much fit well in the theoretical distribution. Here, for example, you have quantile plots that are really horrible. If they deviate too soon from the theoretical distribution, this means that you have a lot of population structure and noise that will make your analysis very difficult. As you can see in our example, we don't have that problem. The results are maybe a bit deflated in the cases of infectivity, ODPS, or disease progression symptoms in a certain case. But for necrosis, we see a very good quantile plot. Why? Because here it's flat, so it matches the theoretical distribution. But at the end, you have this nice tail, nice peak here, which corresponds to the associations with the phenotype. So you want to see this in your plot. That means you will have significant SMPs. Your results are good. Okay. Excuse me. Okay. Another problem that we need to account for is kinship or relatedness. So they have different methods that you can just calculate relatedness in your genotypes. This is a very common thing. It's not that much of a problem, but you have to account for it because, for example, if you are part of a certain family, you will have a higher presence of certain SMPs than, for example, someone from another family. So if they do a Geos study and you have these differences, this again will cause false positives. So by calculating a kinship, we account for this in the model and we avoid this problem. So when we do our Geos study and we want to see exactly which SMPs are significant, which ones do we want to concentrate on, we use different thresholds. The most commonly used one is the Bonferroni correction. So where you, for example, 0.5, you divide it by the number of SMPs in your study, in the Geos study, and then you get a threshold that you want to use. This Bonferroni is a bit too strict in our case because it assumes that all the SMPs are independent when they're tested, but that's not true. In all the genotypes from plants, humans, animals, you have problems like linkage disequilibrium, which means that certain SMPs are not randomly associated in the genome. For example, this comes from meiosis, where you get a spermatozoid from your father and your mother, and they cross-link. So they exchange parts of their chromosome. And we can see here that you have one strictly from your father, second one from your mother, and then you have the last two that come a little bit from your father, the rest from your mother, and vice versa. Now, the probability that some part of the chromosome will cross and will exchange information is calculated because, for example, the ends of the chromosome will have a higher probability of happening. So that will be a random association in the genome. But if you go a bit up around here, there's less probability that this will be exchanged. So these parts, the SNPs here, will get inherited more often. And this is what you want to also think about when you look at your GWAS study. So in our case, we did not go for the Bonferroni correction. We went for the false discovery rate. It was proven to minimize the proportion of false positives, and it's not too strict. So you can still get hits that are important for your GWAS study. And now we can visualize symptoms. This plot here is called the Manhattan plot. Here in the x-axis, you have the chromosomes positions. Arabidopsis thaliana has 5 chromosomes, therefore 5 regions like this. These colors here indicate SMP density at a certain region. And in the y-axis, you have the negative logarithm of the p-value. So basically in here, the smaller the p-value, the higher the peak will be. And this is very nice and easy to visualize. And then you have a nice big peak like this in our study for necrosis. I'm going to concentrate on necrosis because this gave us the best results that we are focusing on further in our study. So in here, you have indicated certain genes that popped up in this study. Now, if you see this peak here is in the same region, which basically means that there is a linkage disequilibrium. This is quite normal in the GWAS study, that these SNPs are linked. So you have to further see which SMP is exactly correlated with your study. Then we come to the point where we want to look at the genetic architecture of your trait. So you want to see what is behind your phenotype. You can do this with the help of a software called GEMMA, where you can perform a Bayesian sparse linear mixed model analysis. This is an analysis that is a hybrid between linear mixed models and sparse regression models, where you can relate your phenotype to many different SNPs at the same time. So it's not a single trait analysis like linear mixed models in previous slides. And this can account for polygenic modeling, which means certain traits might have many genes that influence them, and you can account for this in this study. So BSLMM is really good because you don't really have to know that much about your genetic architecture, and you can use this and it adapts to your data and tells you more about it without you having to think too much about what you should do. So to look at the model behind this, we see that we have presence of the phenotype. For example, here you have a scalar representing the phenotype mean. You also have an NMP matrix. So this is a matrix of individuals, your phenotypes, for example, and genetic markers. The effect size again, where we see how much does one SNP influence a certain trait you're looking at. You also account for errors and random effects in this model. And with this, our results, basically, we were looking at 2 values, so PVE and PGE. PVE in here is the proportion of the phenotype that can be explained with the SNPs that you have in your study. So you genotype for certain SNPs, but you don't get all of them. You catch certain amount in the genome. And with this, you can see how much of those that you have in your study are actually explaining your phenotypes. And the PGE value can tell you how much of your variation in the genotype can be explained with sparse effects. Sparse effects is basically when you have few SNPs that have a very large effect on your phenotype. In many cases, it can be that there are a lot of SNPs that have small effects, but they all influence your phenotype. So when we looked at the genetic architecture between our phenotypes, we could see that in red here, necrosis was very well explained with the genotypes with the SNPs that we have in our study. And looking at the PVE value and at the PGE value, we can see that there's fewer SNPs that explain our phenotype. So few SNPs will explain the whole phenotype, they will be involved in it. not as many as when you look at infectivity. For example, infectivity is not well explained with the genotype, with the SNPs that we have in our study. There are missing parts, and it has a lot of SNPs that are involved in causing this phenotype. And as we can see, for the both viruses, the naive and the evolved one, we have discovered that 2 genes have a very large effect in necrosis. They are the cause of the phenotype. We have a gene AT2G14080, which is a disease resistance protein. The name kind of looks associated with what we are looking at, viral infection. And these genes have been proven to have a role in different infections, not just with viruses before. And we have AT2G14120, which is a dynamine-related protein. Dynamine-related proteins are associated with viral infections. And so just looking at the functions, it seems that these genes are involved and cause most of the phenotype in the necrosis. Okay, so in summary, using linear mixed models, we were able to identify a high peak in chromosome 2, which belongs to the gene AT2G14080, a disease resistance gene. Looking at the genetic architecture, we saw that necrosis is not a very complex trait. It is explained with few SNPs or genes. Compared to DPS, disease progression, infectivity. And it looks like the SNPs that were mapped within genes AT2G14080, also found in the linear mixed model study, and AT2G14120 are major determinants in necrosis development when the plant is infected with Turnip mosaic virus. And I would like to thank people in our lab, Ruben, Paki, Paul, and Santi. And special thanks to our collaborators, Professor Magnus Norberg, Tom Ellis, and Peter Klau from Gregor Mendel Institute. And thank you for your attention.
Thank you, Ana Maria, for the presentation. There's no questions right now, but we can wait a bit. Meanwhile, I want to make a remark and then you to explain a little bit more. All these GWAS analyses, which are, as you have perfectly explained, they are strongly relay in statistical analysis, in complex statistical analysis, but at some point they need to be validated.
Yes.
Okay, I know that you know. Indeed. I just would like you to, I don't know, you have some extra slides with validations or something?
No.
Or just comment a little bit, how do we validate the results of the GWAS?
Well, validating the results, basically you have to go back to genetics and you have to look at which genes have popped up in your analysis, and then do studies like knockout mutants, which is basically where you remove this gene from your plant and then you infect again, and then you see how the disease is progressing, if the symptoms are less severe, or if you don't have any symptoms at all, is the disease progression faster or slower? And then you can compare that with a control plant. Things are a bit complicated because sometimes you have to look at, for example, if you just remove a gene, you look at loss of function mutations, but you can also put a promoter that will cause this gene to be expressed more, and also take a look at that, if this will also affect your infection. And for example, sometimes removing just an SNP and not the whole gene, or mutating it, can have a bigger effect. So we go into the whole CRISPR-Cas mutations, but so it's complicated. But there are ways that you can validate your genes. There's also probably bioinformatic analysis where you can look at, for example, accounting for this SNP in your model and seeing if you still retain peaks or if you have no results at all. This can also be a certain validation of your results. So there's a lot of things you can do. So yeah, bioinformatic analysis would be faster. If you want to go into the whole mutants experiment and SMP mutations, this will probably take longer time. So yeah, you can validate your results this way.
Okay. Just for our more mathematically oriented audience today, you have been presenting the methods which are basically linear model type of modeling. Could you tell us something about other approaches, things like either Bayesian approaches or maybe some even more fashion and modern, like, I don't know, machine learning algorithms, there are something like that out?
So what is most used in association analysis or GWAS studies, linear mixed models. You have variations within models, how they account for certain things, but they all use basically linear mixed models. Sometimes you have general linear mixed models or variations of the same thing. If you want to use Bayesian methods, this is mostly used to look at your genetic architecture, like I mentioned, or to predict phenotypes. When you don't have, if you don't sequence the whole SNPs in your genomic region, so you can use Bayesian methods to predict how certain SNPs might interact with your phenotype. Machine learning methods are being used a lot right now. They're being used before the GWAS study and after GWAS study. So before, you can use them to like perform a normal GWAS study and then let the machine to learn more about your results and the associations and the genetic architecture of the things you're looking at. And then they can predict future steps and, for example, avoid too many false positives. If you want to do it after your GWAS study, after you already performed the association with linear mixed models, you can look at, for example, how correct this is. They can also associate maybe your hits with certain like metabolic networks and connect these results more because, for example, one gene can be involved in a huge network, and then this whole network could have an effect on your phenotype and not just this one gene. So you can look at things like that. It's a growing field. It's interesting, but the machine learning is still kind of in baby steps in GWAS, but they have been doing pretty cool stuff and discovering things. But yeah, so hopefully they continue.
Hopefully. Okay. If there is no more questions from the audience, thanks a lot, Ana Maria.
Thank you.
And thanks to all the participants of these morning sessions. We are going now to make a break until 3 p.m. And then we will have a long talk by Professor Gennady Bokarov